Okay, so most people wanting to use tissue for assessing cancer want to go to surgical pathology. So pretty descriptive term. We're talking about what surgeons remove from patients in dealing with cancer. And I list the common types of specimens that the surgical pathologist receives, but the ones that most researchers are interested in are cancer tissue that's been resected, so entirely removed, or it was excised. Another word, commonplace for excisional biopsy is the breast. Another word for it is lumpectomy. Autopsy also, you have specimens there, but probably less than 5% of our deaths across the country are autopsy today, so it's not much less common than surgical pathology. And what we'll do up in the path lab is we'll cut out pieces of tissue from the specimen that we received and put it into these types of blocks here. And what will happen in the processing then is these blocks, this tissue will be generally in formalin, 10% formalin for fixation for up to 24 hours. And then it'll be permeated with wax in a processor. And in Iowa, in the cancer registry, we have two types of repositories. We have a virtual tissue repository where the specimens that we would be pursuing are housed at pathology facilities around the state, in some cases in neighboring states. And then we just go after the specimens that are needed for an individual study from this repository. And typically it takes more time because we have to request the tissue, and it's up to the laboratory that we send the request to when they find time to pull it. So it typically does take more time. The residual tissue repository is one that we have here in Iowa City. It's over in the University Capital Center. These are specimens that we house, and it consists of tissue blocks and pathology reports. We do not get the slides. So labs elect to hold on to their diagnostic slides in pretty much all instances. So we call that the residual tissue repository because those blocks were slated to be destroyed by the outside path labs, and we intercepted the process. Oh, let's see here. Try again. So as of the middle of this month, our residual tissue repository contained coming up on 600,000 tissue blocks representing almost 70,000 malignant tumors and almost 100,000 pathology reports for about 67,000 cancer patients. The virtual tumor registry repository is even larger. We estimate we have an additional 386,000 tumors out there diagnosed between 1973 and 2018. Now, in the RTR, those 65,799 tumors that we have are distributed across all sites, but the common sites are breast, colorectal, lung, and prostate. These are the major cancers that we see in the state of Iowa and nationwide. And after that, you have bladder, non-Hodgkin's lymphoma, uterus, melanoma. These are all in the top 10. So the residual tissue repository is pretty reflective of what's going on in the state of Iowa. And people can have more than one tumor, so it's not surprising that we have more tumors than we have patients in the RTR. Note up here that these tissue blocks are from 1994 to 2009, reflecting the fact that tissue blocks typically are not destroyed until 10 years after diagnosis. And that's because the laboratories want to maintain their certification by the College of American Pathologists, and a certification requirement is they hold on to tissue blocks for 10 years at least. And then out in the residual tissue repository, we estimate, again, for these same sites that we would have this type of a distribution. And what we have to do in the RTRs, we've got the tissue blocks, so we have to work, well, we don't have to, but we elect to work with the Research Histopathology Lab in the Department of Pathology. And they have histotechnicians there that can take a block that we send over to them, put it on a microtome, and cut a thin section. And these sections are probably 5 microns in thickness here. They typically float these on a water bath and bring up a slide underneath them, put it on a slide, stain it with hematoxin and eosin, and put a cover slip on it. And this is the standard slide that's used for evaluation microscopically of tissue. It's called a H&E, hematoxin and eosin stain slide. So typically we have a specimen, we'll take a sample, we get a tissue block, we get a histologic section. That can be examined. You can identify areas on the slide that we'd like to get a piece of the tissue. You can do special stains on it. You can do immunohistochemistry. A lot of people are interested in that because a lot of the biomarkers that we have, the important ones for cancer, are also immunohistochemically can be detected in the cells, either on the cell membrane, in the cytoplasm, or in the nucleus. You can also do fluorescent in situ hybridization on H&E cut tissue. And people now have the capability of doing DNA and RNA isolation as well. The ability to do this has evolved to the point where it can be done. 10, 15 years ago, people would say, no, the DNA is all chopped up, it's no good, in formalin-fixed paraffin-embedded tissue. But the technique has now evolved to the point where you can get both DNA and RNA, reasonably good quality. And then researchers sometimes are interested in a tissue microarray. I want to show you what happens with that a little bit more. Here they have a tissue block, they have a slide, they put a slide on top of it. Typically this slide is marked because it's been looked at under the microscope. They're matching it up with the tissue block here. They bring a little core thing down here. These cores can vary from a 0.5 millimeter up to 1 or 2 millimeters in diameter. And here they just put it on the slide to indicate where in the tissue block underneath this slide they want to get a core. And they stick that core in there. Then it takes a sample of the tissue right from the area you marked, which may be a specific area of tumor that you're interested in. You can bring that back out and then you can insert it. You see this little button here. You can push that core out into a new tissue block, and this block would constitute a tissue microarray. The value of these is that you can get many of these cores onto one block. So if each of these represents a different patient's tumor, you can see by one test, you can get results on dozens of patient tumor findings. So it's a cost savers for researchers, but it does take a little bit more time to create the block and to get to the point where you have the slide. But once you have this slide, if you have this tissue in here and it's a few millimeters in thickness and you're taking 5 micron sections, which is typically used for immunohistochemistry, you can see that you now have a kind of a gold block here. That's the value of gold to you in the sense that you have many sections you can get from this, which means you can look at many different markers with this and gather information for dozens of patients with a single staining of one slide. And here's another example of which tissue microarray that had many cores put in it. Sometimes these cores are a normal area, sometimes they're different areas of the tumor. Usually these are set up with positive and negative controls as well, so that when you do a stain, you can make sure that it's working based on the positive and negative controls. Now, another thing that's typically done with these blocks is they are labeled. These are labeled with bar markers. Some laboratories have that. Others of them just still manually label them. But they're labeled and they're connected to the pathology report. The pathology report can be linked to the medical record for the person. At UIHC, you can link that information into EPIC, and you can gather information from EPIC to annotate what you have in the block. In the case in the registry, we annotate it to the clinical tumor patient survival information that we have in the registry and annotate the tissue blocks with that information. So the registry variables that we would have available on tissue that people are interested in, patient demographics, age, sex, race. This would be information at time of diagnosis of the tumor, the characteristics, the site of the tumor, in many cases the subsite coding as well, the morphology, whether it was a squamous cell adenocarcinoma, sarcoma, etc., whether it was laterality for paired organs, right or left breast, right or left kidney, extent of disease, was it localized to the organ? Was it spread to regional lymph nodes? Was it spread beyond that to distant metastasis? The surgical procedure that was used, whether it was section of biopsy, and then in some cases we have site-specific factors like HER2/neu, ER/PR positivity for breast cancer, and markers for other types of tumors as well. We're in the process of gathering more of that information now that it's available in the electronic health record, but the process of getting this accomplished is moving, but it's slowly moving. We also collect information on first course treatments, so you know about whether they have surgery, therapy, chemotherapy, immunotherapy. We have this type of information. Then we follow all the patients to death, so we have survival and cause of death. Some people, because cancer is a disease of older people, have also asked to have the data annotated with Medicare information. Medicare has patterns of care data, it has cancer tests and procedures, it has costs, it has comorbidities, so you can add additional information onto what the registry has, and some researchers are interested in that. Now, we're able to do all this largely without patient consent. And the reason why is that cancer is a reportable disease in Iowa, and the registry has been delegated responsibility for the collection of this reportable disease here in Iowa through the Iowa Department of Public Health and in the law of Iowa, the Iowa Administrative Code. And with HIPAA, a registry like the Cancer Registry was designated as a public health authority, which means that we were exempt from HIPAA requirements for its required data variables. And the paraffin block repository is part of the registry. It is approved by the UIRB. So the UIRB said you can share this material with researchers, but every time you do it, it's going to require a separate IRB approval. And furthermore, every time we've done this, they've granted a waiver of informed consent, in part because we've de-identified the data. So you don't get names, social security numbers from us, but you can get age and race and sex. And some of this information that we can let out is being tightened up a little bit now with some of the changes to the administrative code, but it's still pretty good for meeting the research interest of investigators. Now, typical steps to obtaining tissue is usually we'd like to see a brief proposal, what it is you want to do. There's a request form out on the website because we're part of the SEER Residual Tissue Repository Biospecimen program. So that form could be filled out. But it typically comes down to let us know what it is, what are your specific aims, what do you feel you need. We handle most of this by email, and then as we exchange ideas and get comments on the proposal, we generally will have a teleconference. We'll talk about what we can provide. Sometimes it's not what you had expected based on the proposal. Sometimes it's more than what you had expected. So typically the specific aims get more focused as a result of these teleconferences. And then other questions and concerns can be addressed, including funding. It does cost money, some money to get this tissue. So you have to figure out a way to pay for that. And a lot of people pursue grant funding for this purpose. So sometimes we're working with a proposal and it'll be well over a year or more before we find out whether it's really going to move forward because of it took that long to pursue getting funding. We also usually hone the eligibility criteria based on that brief proposal so that sometimes you find out we have more cases than what you thought, so you can be more specific in your specific aims. We typically will provide a preliminary data table with the number of eligible cases by key variables that meet your eligibility criteria. We'll give you a case count of how many cases we will pursue. It doesn't mean we'll be able to get tissue on every one of these because we still don't know exactly how much tumor tissue is in a block until we're able to review an H&E slide, and we're not going to do that until there is some funding for the project. Typically, what I'll do in projects I'm involved with, we have your proposal. Sometimes that's a grant proposal. But we have to figure out what we're going to do on our end and what you're going to do on your end. So what we're going to do on our end goes into a technical protocol that we develop for our own use here. We'll pursue that with our IRB to get approval for the research we're doing at Iowa in regard to your proposal. And then we can put a budget to that as well. And then the funding, as I said, needs to be obtained. And after that, there needs to be approvals, IRB, If we're going to be transferring material to the other institution, we need a material transfer agreement that's required by sponsored programs at the University of Iowa. And the Iowa Department of Public Health, if you want cause of death, you have to get approval from them as well for the release of that information. Typically then what you'll get from us is the tissue that you requested from the blocks, and you'll get a de-identified path report that goes with those. And sometimes people want an H&E stain slide as well. We'll send that. Increasingly, people are starting to image those slides so that they have those set aside in a computerized format. And the study goes on then. So we're working on the front end. Sometimes people want us to send them the tissue block because they're going to do something special. We can do that, but in instances like that, we want to get reasonable turnaround time, particularly in utilizing the virtual tissue repository, so that those participating labs on the outside don't get irritated with us and say, we're not going to work with you anymore. That would be bad for you. It would also be bad for future researchers if we can't go to that lab to get any blocks. So here's some examples of studies that have gone on. We've done tissue on liver cancer, on breast cancer, on HPV-associated cancers, ovarian cancer, pancreatic cancer, a rare tumor, sebaceous carcinoma, but we were looking for viral etiology of these sebaceous carcinomas. You can see a lot of our researchers are not at the University of Iowa. There are some examples of where they are, but many of them are coming from the National Cancer Institute and from other universities, the Fred Hutchinson Cancer Research Center and Memorial Sloan Kettering Cancer Center. And we use RTR and VTR for some studies, sometimes just the RTR, other times just the VTR.